Hi folks, okay, this is the second talk on phylogenetics in practice. After the first one, which focused on phylogenies, what they mean, and how to infer them, this one's going to focus more on population genetics and how we can use population genetics theory in practice to learn things about the demographic history, for example, of sequences sampled from within a species. So if you enjoyed the talk on phylogenetics and were familiar with a lot of the concepts, then you probably will like this one less, because it's very different in the sense that we're going to focus more on the theory underlying the different models, and it's going to be quite different in flavour. So let's really start with the theory underpinning the use of sequence diversity to learn about population histories. Let's talk about molecular clocks and neutral evolution. Although I brushed over this a little in the talk on phylogenies, when converting phylogenetic branch lengths to calendar time, one has to make assumptions, and the commonest and probably simplest assumption is to assume a strict so-called molecular clock. What that means is that there's a constant rate of substitution, the change of nucleotides from one to another, along the phylogeny. Of course, the question is it reasonable to assume a molecular clock, and if so, what are the justifications? So population genetics provides some insights. In population genetics, typically one has to make a range of assumptions, and so we're going to consider an idealized population which captures the typical assumptions. Firstly, that it's large, that it's constant in size over time, that the population is well mixed, it's not structured. And based on those assumptions, we can start to produce mathematical models that tell us about quantities of interest. And when I say we, what I mean is people like Muthu Kimura. Okay, so the first quantity that one wishes to understand is the substitution rate. Now, if you take substitutions back to the level of within populations and apply population genetics to predict how quickly sequences will change over long periods of time, then the quantity of interest is known as the fixation probability. Fixation means that starting from a new mutation, which is at extremely low frequency, what's the probability that it goes to become essentially ubiquitous within the population, so that it sweeps from a frequency of close to 0 to a frequency of essentially 1. That's fixation, and the probability of fixation, although I'm not going to tell you how it's derived, depends on some key quantities which we can understand fairly simply. So this equation here gives you the probability of fixation. So a new mutation, which starts off at frequency f, has this probability, and it depends on only two quantities: f, its current frequency in the population, and then gamma. So gamma is the so-called population scaled selection coefficient, and in turn it depends on a small number of things that we can easily understand. So the first is the ploidy of the population. So for each individual in the population, how many copies of the gene does it carry? For diploids, p is 2, and for haploids, which will be most pathogens, it will be 1. N is the population size, the number of individuals. And then S is the relative advantage, survival or reproductive advantage, to an individual that carries that particular mutation relative to the wild type. So 1 plus S is the relative fitness advantage of the mutant. And roughly speaking, that means in a particular generation, if for every 1 offspring that the wild type is expected to produce, the mutant is expected to produce 1 plus S offspring. If you look at the graph, it shows you, given the current frequency of the mutant and the strength of selection favouring the mutant, which is quantified by this compound parameter gamma, tells you the probability of ultimate fixation. And so if you take the grey line in the middle, that's the neutral line, so that's a mutant which is neither beneficial nor disadvantageous relative to the incumbent wild type allele, then you can see that, and this is a key insight from population genetics, that the probability of fixation is simply equal to the current frequency of that allele in the population at any one time. What that means is that when the allele first arises, for example, it's present in one individual, then the probability of fixation is extremely low. and it's on the order of 1 over the population size. Whereas when it's got 50% frequency, then there's equal odds that it will fix or ultimately be lost from the population. Now, it's no stretch of the imagination that advantageous mutants have got a higher probability of fixing, of sweeping through the population. And the stronger their selective advantage, the more likely they are to sweep through the population. So with a population scaled selection coefficient of gamma equals 1, there's a modest increase in the chances of fixation relative to the neutral case in grey. And when you get up to a population scaled selection coefficient of gamma equals 10, then there's a considerable increase relative to what you'd expect under neutrality. And the situation is mirrored for new mutations which are disadvantageous. There's a few key points to bring out straight away. So the first is that the strength of selection, in a sense what natural selection sees, depends on two quantities. It depends on both the inherent advantage of that allele, which is represented by S, the relative fitness, but also the size of the population, which I've called N. And the reason for that is because selection is more efficient or efficacious in larger populations. So larger populations are able to more efficiently sort the wheat from the chaff, and they can more rapidly spread advantageous mutations and more effectively remove deleterious mutations. And so for a particular for a mutation with a particular size compared to another one which is 10 times more advantageous, if the mutant which is 10 times more advantageous arises in a population which is 10 times smaller than the less advantageous mutant, then those two things end up cancelling out, and they've both got an equal chance of finally making it to fixation. So that's the first key point. Selection depends on both of those two parameters, the size of the population and the intrinsic advantage of the mutant. The second really important thing to notice, and I'll point to it here, is that when new mutants are at low frequency, whether they're advantageous or deleterious, the probability of fixation is low. And what that tells you is that chance is really important during the early days of a new advantageous mutation. And many, even considerably advantageous mutations, are lost simply due to random chance when they're at low frequency. And that process is known as genetic drift. There's some useful approximations that one can make and some famous results in population genetics. The first one that I'll show you is for a mutant which confers an advantage which is not too large, so much less than 1, then the probability of ultimate fixation in a large population is approximately equal to twice its selective advantage. And the second famous result, which I'm going to show you, which is even more important because it underpins Muto-Kimura's theory of neutral evolution, is that the probability of fixation of a neutral mutation equals its initial frequency. So if P is the ploidy and N's the population size, then it's 1 over Pn. And that just reflects that straight grey line in that graph. And this is why that's important, because in trying to understand what rates of substitution are likely to look like at very, very deep timescales, for example, geologically relevant timescales over the whole tree of life, then one is able to argue from a bottom-up population genetics approach that substitution rate should be constant, and this is why. So for mutations which are neutral, and I'll come back to exactly what neutral means, but for mutations which are neutral, they don't confer a strong advantage or disadvantage, then we've already covered what their probability of fixation is, given that they've arisen, and what's the rate at which they arise. Well, if we assume that there's a constant mutation rate, which I've called here mu, then because there are Pn copies of any particular gene and therefore any particular nucleotide in the population, then the total rate at which those mutants are generated every generation is P times N times mu. So the substitution rate is then equal to the rate at which new mutations arise by mutation times the probability that each of those goes on to fix. And the key thing and the key insight of neutral theory is that the population sizes cancel and the substitution rate winds up being simply equal to the mutation rate. So if the mutation rate is constant, neutral theory predicts that the substitution rate will be constant. Okay, so what exactly do we mean by neutral mutations? And what about beneficial and deleterious mutations, which of course are very important over long periods of evolutionary time? They're particularly important because they explain the diversity of adaptation that we see between different organisms occupying different niches. Well, contrary to a common misconception, Kimura's neutral theory allows for beneficial mutants and deleterious mutants, but it assumes that both of them are rare and fleeting. And why does it do that? Well, firstly, it assumes that they're rare because most organisms are already well adapted to their environments, and so large advantageous mutants are not likely to arise. Second is that if their selective advantage is large enough, then they're likely to sweep through the population and go to fixation quickly. And indeed, if they don't go to fixation because of loss due to drift, that process is also likely to occur more quickly than for neutral mutations. That's a non-intuitive result. So beneficial mutations are assumed to be rare and also fleeting. Likewise, the substitution of deleterious mutants is assumed to be rare and fleeting, and that's because, like beneficial mutations, their trajectory to fixation or loss is expected to be quicker than for neutral mutations. And in this particular case, although deleterious mutations are not likely to be rare in terms of their origin, because if organisms are already well adapted, then it's quite likely that any mutation will be deleterious. The substitution of those mutations is very unlikely, and for that reason, substitution of deleterious mutations is rare. And so what these arguments suggest is that actually, in terms of the vast majority of substitutions that one observes over long periods of evolutionary time, although particularly adaptation and adaptive mutations do occur, because they're rare, because they're fleeting, then overwhelmingly molecular evolution occurs in a neutral manner, and that predicts a strict molecular clock if the neutral mutation rate is constant. And that's a key result, because what it tells you is that it doesn't matter how populations change, it doesn't matter if they're bigger and therefore better able, more efficacious, better able to select advantageous mutations, better able to weed out deleterious mutations. It doesn't matter if they're small and the opposite applies, because those things cancel out for the vast majority of mutations, which are assumed to be neutral because of the arguments above, and therefore that predicts a molecular clock. Okay, so that's neutral theory, and I think neutral theory is an important prerequisite to coalescent theory. The coalescent theory is fundamental to exploiting patterns of diversity within populations to understand the evolutionary history of the species. I'm going to explain you why that is. Modern population genetics is largely framed in terms of coalescent theory, and it started in Oxford with John Kingman in the Department of Statistics with a seminal paper in 1982. The coalescent is a bit of a mystery to many people, but actually all it is a statistical distribution. It's a statistical distribution in the same sense that a normal distribution or a binomial distribution is a statistical distribution, but instead of applying to continuous or discrete quantities like those two distributions, what the coalescent does is it describes the likely distribution of trees, including branch lengths, between individuals within an idealized population under the assumptions of the standard neutral model. Hence the importance of neutral theory. And as explained already, the idealized population is large, seem to be constant over time, well mixed, and to bring in neutral theory with negligible fitness differences between individuals. So the relatedness structure or phylogeny or genealogy between members within a species, when the species can be approximated by these assumptions, is modelled by the coalescent. So it's a tree in the absence of recombination, otherwise it's a network. And it's useful because not only is it tractable from a statistical perspective, and not only are its assumptions defensible by appealing to neutral theory, but it provides a baseline model on which many extensions, including, for example, coalescent with selection, have been built. So this is an example coalescent tree or genealogy or phylogeny within a species. And the reason I keep emphasizing within a species is because these assumptions about how the population acts are really key to the validity of this statistical model. And they're simply not likely to act in the same way when you consider multiple sequences taken from different species. Okay, so the coalescent model, I told you it's like a normal distribution, but it's for trees, including their topology and branch lengths. And in this tree, what I've done is I've taken, let's see how many there's, 4, 5, 6, 7, 8, 9, 10. Are there 10? I'm just going to count that. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 tips in this tree. And what I've done is I've split the tree into so-called epochs. So I'm assuming that all of these different sequences within the population were sampled at the same time, the present, which is labelled 0, and then the axis on the left measures time going backwards. And you can see that the blue dashed lines separate the tree into so-called epochs, during which the sample had the same number of common ancestors. So the final epoch before the universal common ancestor of the samples has 2 lineages, then the one more recent than that has 3, and so on. The first result from coalescent theory under its assumptions is that every tree topology is equally likely, or formally it's uniformly likely. And so really the tree is characterized by these epochs, or the intervals between successive coalescent events. And under the coalescent, they follow a simple distribution called the exponential distribution, and it has a mean which is expressed as follows. It's expressed in terms of the ploidy and the population size. You times those two things together and it tells you how many genes there are, how many copies of a gene, or indeed a single nucleotide in the population. And k, which is the number of lineages within that epoch, within that interval. And just to come back to a point that I made in terms of the interpretation of phylogenies or trees in the previous talk, is that each of these nodes in the tree, which in this context are called coalescence events, are events at which 2 or more sampled sequences in our sample find the most recent common ancestor. But equally, if you turn it around and think, what does it actually mean biologically forwards in time? They're reproductive events in which a particular cell left 2 descendant cells, both of which went on to leave descendants that were sampled by us. So they're reproductive events, but they're conditional on leaving descendants, both of which had lines which were then ultimately sampled by us. And that's what a coalescent event is. So take a look at the general shape of that tree, because I'm going to show you what it looks like when you alter the assumptions a bit and allow for changes in population size, in particular population growth. Compared to a constant size population on the left, a coalescent tree, or the genealogy of a random sample of individuals within a population, is compressed when you have population growth. Now, I just want to talk about that random sampling assumption because I haven't really emphasized it previously, but if the population is well mixed, then one assumes that one's sampling randomly from essentially a homogeneous population. And of course, as with any of these assumptions, if that's not true, then one has model misspecification, which can then mislead inference. Okay, so there's a signature in terms of the tree, which is left by demographic processes, in particular changes in population size. And in the case of population expansion, then what you see is a general compressing of the tree. And not only a compressing of the tree, but the branches closer to the root, which tended to be longer, are actually more equal compared to branches close to the tips of the tree than you would expect in a population which is constant size. Every tree topology is still uniformly likely. As I've said, the intervals between successive coalescent events are compressed, and the compression gets stronger as the population contracts further back in time. And so that reduces the difference in mean coalescence intervals near the tips versus the root of the tree, and the tree is said to become more star-like. What use is this? Because actually one doesn't directly observe trees, one observes sequences. And the key point is that one can infer the tree, and by inferring the tree, one can interpret patterns of genetic diversity in terms of demographic changes over time. And so one can really kind of read the tea leaves, and by interpreting sequences in the right way, one can learn things which are not immediately apparent. For example, that the population has expanded, moreover how quickly it expanded, and when it started to expand. And so one's able to estimate growth rates from sequences via the trees that we can infer from them. And typically this is done in a Bayesian framework. So now I need to explain to you what Bayesian phylogenetics is. And to do that, I'm going to come back to this court case example with which I explain the principles of maximum likelihood. So the ingredients of a maximum likelihood method are parameters, a statistical model and data. And in our particular case, the parameter of interest was whether the defendant was guilty, theta equals 1, or not guilty, theta equals 0. Obviously, that's just a formal device. We could call them whatever we want and give them whatever numbers. It's just a binary outcome. The statistical model is a crucial bit. And in this case, it considers how frequent are shoes matching the print among the suspect population. And of course, the statistical model is tailored to the data, and the data in our case was that the defendant has shoes matching a print found at the crime scene. Of course, the data could also have been that the defendant does not have shoes matching a print found at the scene of the crime. But given that he did, we evaluated the odds that the defendant was guilty versus not guilty as 1 over f. And so what that means is that if f is less than 1, then the defendant's more likely to be guilty than not guilty based on an interpretation of this evidence using this particular model. So you're a juror in court and you're presented with this data. It probably wouldn't be presented in exactly this way. Would you find the defendant guilty or not guilty? Well, Bayesian inference is applied in a slightly different way. Let me just go back a second. Because you might think that actually, if f is quite high, so f might be 90%, so if actually lots and lots of people in the population of possible suspects have trainers matching the particular footprint found at the scene of crime, then you would be right in thinking that actually the evidence is not particularly strong in favour of them being guilty or not guilty. So the maximum likelihood estimate, which is our best guess, would really be perhaps a little harsh and draconian in this particular case. And what are we missing here? Well, Bayesian inference has some answers to that question. So Bayesians base inference not on likelihoods directly, but rather on so-called posterior probabilities. And they account not just for data through the likelihood, but also for prior information. And so in our case, the prior information would relate to the parameters of interest, which is whether the defendant is guilty or not guilty. You can represent this formally in terms of these odds. So on the left of the equation are the odds that the defendant is guilty or not guilty given the data. And on the right-hand side, you've got two quantities. So on the far right is the likelihood ratio, which is what the principle of maximum likelihood uses in order to make best guesses. I shouldn't really call them guesses, but you know what I mean. So that's the likelihood ratio on the right-hand side. But then we've got this extra term, and that's the prior odds that the defendant is guilty or not guilty. And so the difference between the Bayesian approach and the maximum likelihood approach is in the use of these prior odds to modify the raw interpretation of the data in order to arrive at posterior odds, and on that basis decisions can be made. So what might these prior odds look like in the court case example? And I want you to think about how you might, let me point to this particular term, these prior odds, what might these look like? So let me remind you that the likelihood ratio is 1 over f. So we've assumed that the probability that there are matching footprints given that the defendant is guilty is 1, and given that he's not guilty, f. Now, what are the prior odds that that individual is guilty or not guilty? So if you didn't see any information, before you had any information, you don't even know that the police suspect this particular individual, and you don't know that he has shoes matching the footprint at the crime scene. Before you have any information, what would be your prior odds that this particular individual, taken from the population at random, or maybe the population of suspects which are known to the police, is guilty? Well, I think a reasonable assumption might be that he's equally likely as anyone else within that population. And so if the size of that population is n, then the prior odds that he's guilty or not guilty are 1 versus n minus 1. So 1, because it's him, or n minus 1 because it's anybody else. So that's why that ratio is 1 over n minus 1. Assuming that everyone in that suspect population is equally likely before receiving any information. So where is the maximum likelihood estimate of theta, the most likely scenario, the interpretation of the parameter that best fits the data, tells us that the defendant is guilty unless everybody else in the population also has those shoes, not probable. The Bayesian interpretation is somewhat different. So the most likely scenario in the Bayesian's view, which is known as the maximum a posteriori estimate of the parameter theta, would equal 1, guilt, only if f exceeds not 1, but 1 over n minus 1. And let's assume that population is reasonably large, maybe there's 100 individuals. So f needs to be, I said exceed, but what I mean is it needs to be smaller than 1 over, say, 99. So that's a big difference. The maximum likelihood approach would consider guilt to be the most likely explanation unless everybody in the population has shoes, whereas the Bayesian approach would consider the defendant to be guilty and that to be the most likely explanation only if 1 out of every 99 people in that population, or fewer, have shoes matching the footprint found at the scene of the crime. It's a fundamental difference, and it's because it's taken into account the prior odds that this particular individual was guilty before observing any of the data. Like maximum likelihood, there's extensive theory that motivates the use of Bayesian inference. So Bayesian estimates are, obviously they're biased by the prior, and that's a good thing if your prior contains reliable and useful information. They're less noisy than maximum likelihood estimates, particularly when you don't have much data, because the prior adds information. They're increasingly independent. The Bayesian estimates are increasingly independent of the prior. As you get more and more data, ultimately the data overwhelms the prior. But because they're useful for small samples, they're not reliant on so-called large sample asymptotics for the construction of things like hypothesis tests, p-values, and quantifications of uncertainty. Depending how you construct your point estimates and credibility intervals, which is the Bayesian word for confidence intervals, then you can also achieve this property of invariance to transformation. But of course, the big caveat with the Bayesian approach, and the big downside as seen by many, which is that if you don't have reliable or well-informed prior information, then you need to find a different way to identify and justify some sort of prior, and these sorts of approaches refer to those kind of default priors as reference, objective, or non-informative priors. And that's a large area of research, and it's probably the most contentious issue of Bayesian inference, which is how to specify the prior when you don't have good prior evidence or a reasonable justification for your assumption, as in the court case scenario. Okay, if you're mathematically inclined, you might want to dwell on this slide, and if you're not, you might want to skip over it quickly. Bayesian inference is based on the posterior distribution, and that's what all downstream decisions are based on, such as obtaining point estimates and credibility intervals. And so here I'm just showing you the derivation of the posterior probability of guilt versus not guilt in the court case example. So given the data, you have this expression here. So this is the posterior probability of guilt given the data. You apply Bayes' rule to this, hence Bayesian inference, and you get an expression like this. And if you plug in the numbers from our assumed model and prior, then you end up with a particular expression. Then the probability of not being guilty given the data is 1 minus that. Now, to look at it visually, what you can see for an example of a suspect population of 1,000 individuals and a frequency of trainers within that population of 1%, then you can see that the posterior probability given the matching footprints of guilt is much lower than the posterior probability of not being guilty. Point estimates can be defined in different ways. I'm going to skim over this in the talk, but if you're interested and you want to try to pick apart the details, then you can dwell on this slide and maybe use it as a starting point for looking into more formal texts. So in terms of point estimates, you can take this posterior distribution, which is represented in the picture below, and you can summarize it in different ways. And this is an extremely simple posterior distribution because the parameter can only take two possible values, 0 or 1, representing not guilty or guilty. But even so, these principles apply more broadly to ordinal values. So you can take the mean, the median, or the modes. These are all different averages, and you can write down different equations. In a sense, the mode corresponds most closely to what you do under maximum likelihood. So you find the value of the parameter which maximizes the posterior probability, and that's known as the maximum a posteriori estimate, or the MAP estimate. And that's what we saw on the previous slide. Likewise, there's different methods for obtaining credibility intervals, which is the Bayesian equivalent of confidence intervals. They can be defined as any interval over that posterior as containing 95% of the probability. And the common ones that you'll see are the so-called 95% highest posterior density interval, HPD. You can work through this simple example and calculate what that would be. And the equal-tailed interval, and again a 95% credibility interval in this case would take the 2.5 and 97.5% tails of the distribution, those percentiles, and calculate what those are. And in this simple example, you can work through them, but it's probably a bit more natural to think of those in terms of histograms. It's a bit of a whistle-stop tour, but you might get more of a sense of intuition for what all of this means in the practical, where we're going to be using Bayesian inference when we apply BEAST to learn about demographic signal in the Zika virus outbreak. So to summarise Bayesian tree builders, they're slow. They're usually slower than maximum likelihood approaches, which are in turn much slower than distance-based methods. Their usage requires not only mathematical models for the data, but now also for the priors, and it requires specialist software. On the plus side, there are theoretical performance guarantees, which are not the same as for maximum likelihood methods. They're quantitative, so they're able to assess uncertainty. The assumptions are explicit, and again, they exploit all information like maximum likelihood methods. So let's turn to BEAST, which we're going to use in anger in the practical. BEAST is short for Bayesian Evolutionary Analysis Sampling Trees. There's a reference paper. And so BEAST is one method which applies Bayesian reasoning to infer phylogenies, and in particular it's particularly appropriate for sequences sampled within species, subject to the phylogenetic assumption that there's no recombination. And actually, that assumption is often reasonable for pathogens, which are frequently analysed using BEAST. And that's because even though probably most pathogens are capable of recombination, often the rates of recombination are relatively slow, and during outbreak scenarios, events are happening so quickly that recombination can be ignored. And that's really an empirical question, and that's why it's essential to check whether there's any evidence of recombination within the sample using methods, and there'll be an example of that in the practical. So when applying Bayesian approaches to learning about trees, a key question is what prior distribution should we assume for the tree and its branch lengths? And to start to bring together the different strands of this talk, the coalescent has advantages as a prior. So the coalescent, as explained, is a statistical distribution for trees, so it can act as a prior on trees, their topologies and branch lengths. It's motivated by bottom-up population genetics theory. In the coalescent, the tree is forced to be rooted with the tips at known sampling dates, and these constraints improve interpretability of trees. And there are many extensions to the standard coalescent, such as population growth, which can be used, and then the parameters of such growth rates can be jointly estimated along with the tree, which carries through all uncertainty, including uncertainty in the tree itself, into the final conclusions and interpretation that we make. But the coalescent is not the only choice. Other priors are available for trees. As I've mentioned previously, the standard coalescent is not appropriate for mixed species data. In particular, the assumption that the population is a single well-mixed homogeneous entity clearly doesn't apply when you've got multiple species. BEAST also implements the Yule process for rooted data trees, and Mr. Bayes implements other priors for unrooted trees. And these are more descriptive models. They're not based on a bottom-up population genetics model, really. There's a reference for Mr. Bayes. Not only do we need a prior for the tree, its topology and branch lengths, but priors are needed for all other parameters, such as the clock rate, growth rate, and the transition-transversion ratio. Okay, so let's talk about what BEAST actually does in practice. What it doesn't do is find the MAP estimate, the maximum a posteriori estimate of the phylogeny and its branch lengths. And the reason that it doesn't do that, one of the reasons, is because that would require a search strategy similar to maximum likelihood methods. Another problem with MAP and maximum likelihood point estimates of a tree is that they don't quantify uncertainty. So in the maximum likelihood and also distance-based methods, one can use methods such as bootstrap for approximating confidence intervals, but instead what Bayesians do is that they use the posterior distribution both for obtaining point estates and also credibility intervals. So the challenge, as in maximum likelihood methods, is to explore all the possible trees so from the Bayesian setting to explore the posterior distribution of the tree, its branch lengths and the other parameters. And to do that, BEAST uses a method that's very widely used and is worth knowing about if you don't already, which you may well do, which is Markov chain Monte Carlo, or MCMC. So let me try to motivate what MCMC is, because it's a little complicated, and so the question arises, what is the need for the complexity? Well, for some simple models, sometimes it's possible to write a formula for point estimates and indeed credibility intervals. And an example of that is the court case where we were able to write down everything using equations. More often than not, with sequence data, and indeed many statistical models of any complexity, that's not possible. But sometimes the parameters are simple enough, for example, a single continuous parameter, they're simple enough that that one can calculate point estimates and credibility intervals by relatively fast techniques such as numerical integration, optimization, or root finding, which I'm not going to explain. But very often neither of those approaches is possible. And so an alternative way to obtain point and interval estimates is by simulation. For example, suppose that you could sample, i.e. simulate, 1,000 draws from the posterior distribution, then one could simply approximate the posterior mean as the sample mean over those 1,000 simulations and approximate the 95% credibility interval from the sample percentiles, i.e. using the equal-tailed credibility interval. But the problem is that direct simulation may not be possible, and usually when the first two approaches aren't possible, then direct simulation is not possible either. And this is where MCMC comes in. So it's a somewhat complex method which addresses a pretty complicated problem. So MCMC starts from an arbitrary parameter value, or combination of parameter values if you've got multiple parameters, as you will in the phylogenetic setting, and it proposes changes to the current values, and those changes are either accepted or rejected depending on whether they improve the posterior or whether they make it worse. So the posterior, by improve it, the posterior includes both the likelihood, so the probability of the observed data given the parameters, but also your prior on those parameters. Formally, a random walk is produced by MCMC, and eventually it's long enough that you can thin it down to an approximately independent set of draws from the posterior and then implement the direct simulation approach to finding point estimates and confidence intervals. So this is an example of an MCMC as applied to the court case example. And so on the left, what you can see is that you've got theta, and that can only take two values, 0 or 1. And you can see this random walk over 100 iterations of the MCMC. It spends some of the time in state 0, which means not guilty, and some of the time in state 1, which is guilty. And you see this autocorrelation. So if the parameter has a particular value in iteration 1, it's more likely than not to have the same or a similar value in iteration 2 because you make local updates to the parameter. The idea is that you run this for long enough that you can summarize the chain in a histogram or a bar plot, as you can see on the right, and we can compare the MCMC for this example to the explicit mathematical formulas in green to look at the accuracy. And you can obtain standard errors for the MCMC-based estimates of posterior probabilities or means or confidence intervals. And you can see that I've run, in this particular example, over 1,000 iterations rather than 100 iterations. I've run it for long enough that the MCMC, or at least its standard error, is within the direct mathematical formula quantification of the posterior probability of guilty or not guilty. And one can do things like, say, have I run it for long enough by calculating quantities such as the ESS, the effective sample size, which in this case was 250. Now, applying MCMC to this very simple example is like trying to crack a nut with a sledgehammer. It's not necessary, but the idea is to show the relationship between the sort of approaches that are tractable in very simple methods and these more complex, heavy-duty, industrial type approaches for cracking some of these more complicated problems. Okay, that's the end of the 2 talks, the introduction to phylogenetics in practice. Here are some of the references relevant to the second talk, and now you can crack on with the second part of the tutorial.